For centuries, humans have searched for cures to diseases. We've explored the natural world from plants to minerals looking for substances that could heal us. This quest for new medicines continues today, but with powerful new tools at our disposal. Modern drug discovery is a complex and challenging process. It involves identifying promising drug targets, screening vast libraries of molecules, and conducting rigorous clinical trials. This process can take many years and cost billions of dollars. To accelerate the discovery of new medicines, scientists are increasingly turning to computational methods, such as virtual screening. Virtual screening uses computer simulations to identify molecules that are likely to bind to a specific drug target. This allows scientists to quickly and efficiently screen millions of molecules, saving both time and money. Virtual screening has already led to the discovery of several important drugs, including HIV protease inhibitors and anti-cancer drugs. As computational power continues to increase and algorithms become more sophisticated, virtual screening is poised to play an even greater role in drug discovery in the years to come. Imagine searching for a key to open a specific lock. You could try every key in a giant keyring, but that would take a very long time. A faster approach would be to use your knowledge of the lock's shape to narrow down the search. Virtual screening works in a similar way. Instead of physically testing millions of molecules in a lab, scientists use computer simulations to identify molecules that are most likely to interact with a specific drug target, like a protein involved in a disease. This digital shortcut saves enormous amounts of time and resources. There are two main types of virtual screening: ligand-based and structure-based. Ligand-based virtual screening uses information about known active molecules to find similar molecules that might also be active. It's like finding keys that look alike, hoping they open similar locks. Structure-based virtual screening, on the other hand, uses the three-dimensional structure of the target protein to identify molecules that fit its shape and chemical properties. This is like using a blueprint of the lock to design a key that fits perfectly. Imagine building a Lego model by finding the right bricks that fit together. Structure-based virtual screening works in a similar way, but instead of Legos, we're dealing with molecules and proteins. In SBVS, we use the three-dimensional structure of the target protein, often obtained using techniques like X-ray crystallography or cryo-electron microscopy, to guide our search for potential drug molecules. We can think of the protein as a lock and the drug molecules as keys. The goal is to find molecules that fit snugly into a specific region of the protein called the active site, where they can block or modulate the protein's function. These molecules are like keys that fit the lock perfectly and can turn the mechanism. SBVS involves several steps, including preparing the protein and ligand structures, docking the ligand into the protein's active site, and analyzing the results to identify the most promising candidates. This process is computationally intensive, but it allows us to explore vast chemical libraries and identify potential drug molecules with high precision and speed. Section 4. Setting up your virtual lab with Python. Python has become a popular language for virtual screening due to its ease of use and the availability of powerful libraries for scientific computing. Setting up a Python environment for virtual screening is relatively straightforward. First, you'll need to install Python on your computer. Then, you'll need to install some essential libraries, including RDKit for handling chemical information, NumPy for numerical computations, and PyMOL for visualizing molecules and results. RDKit provides a collection of cheminformatics tools for working with molecules, calculating properties, and preparing them for docking. NumPy is the workhorse of numerical computing in Python, providing efficient arrays and functions for working with large datasets. PyMOL is a powerful molecular visualization tool that allows you to view and interact with three-dimensional structures of proteins and molecules. With these libraries installed, you'll have the basic tools you need to start performing virtual screening experiments in Python. Section 5. Preparing molecules for docking, a digital makeover. Before we can dock molecules into a protein, we need to prepare them digitally. This is like cleaning and polishing a key before trying it in a lock. Molecules come in various formats and may have missing or incorrect information. We use tools like RDKit to convert molecules to a standard format, add missing hydrogen atoms, and generate three-dimensional coordinates. This ensures that our molecules are in the best possible shape for docking. During preparation, we also optimize the molecule's geometry, ensuring that the atoms are in their most favorable positions. This is like finding the most comfortable way for the key to fit in the lock. Once our molecules are prepared, we can save them in a format suitable for docking software, such as AutoDock Vina, a popular open-source program for molecular docking.